So now we're going to talk about nucleotides versus nucleosides and how we start putting these pieces together. So we talked about the structures of DNA and RNA, the difference being in the sugar, but also the nitrogenous bases, where RNA has uracil, whereas DNA has thymine. So if we take a look, here we have our nitrogenous bases. So again, how these are put together is if you take the base, right? So here's our nitrogenous base by itself. When you take the nitrogenous base plus the sugar, so this is the base, the nitrogenous base, plus the sugar, meaning either ribose or deoxyribose, you make a nucleoside. So you remember sugar for side, okay? Finally, if you want to make a nucleotide, that is a base plus the sugar plus a phosphate group, okay? Now this can be a single phosphate group, monophosphate, or 2-diphosphate, or triphosphate. So for example, you may have heard of something called ATP. If we break this term apart, we have adenosine, which is, again, adenosine is our nucleoside. So we have a ribose, okay, plus the sugar, or excuse me, the base, the adenine base, and the ribose sugar, adenosine triphosphate, or 5' triphosphate. So there are 3 phosphates put together, and the 5' comes from the fact that, again, we're always going to attach the phosphates to carbon number 5 on that ribose sugar. Okay, so you can see in the slides there we have the names of the nitrogenous bases: adenine, guanine, thymine, cytosine, uracil. Then we modify them: adenosine, guanosine, thymidine, cytidine, uridine, to give us the nucleoside. And then when we add now the nucleotide, the phosphates, we add the word of the nucleoside plus, and then we add 5' diphosphate, monophosphate, triphosphate, depending on how many we have in there. So that's how we can look at naming these nucleosides and the nucleotides. So again, putting that all together, we have our base plus our sugar. So without drawing the entirety of the base, we can look at making those 5' nucleotides. So again, if we are looking at, again, carbon number 2 in OH group, so this is ribo, right? We have H, and we have a nitrogen with the rest of the base, right? Because the beta nitrogen linkage there. Now, our phosphate goes on carbon 5. So 1, 2, 3, 4, carbon 5 up here. So CH2, now we have the oxygen that normally is the OH group, okay? That will now be connected to a phosphate. Now, is it 1 phosphate or 2 or 3? Tells us if it is mono, di, or tri. So we call it the 5', okay, because DNA is actually what we call directional. So we link different nucleotides together via the 5' and the 3' carbon, so they go all in order. So if you take a look at a full structure that we have, for example, here with what we call AMP, adenosine 5' monophosphate. So you can see here we have the nitrogenous base, adenine. We have the ribose sugar. Here's the 5' carbon. And here we have a monophosphate, and it's an ester because remember, this is an ester linkage, double bond O to another oxygen, but it's a phosphate, so it's a monophosphate. We could also have a diphosphate if we put a second phosphate here, or a triphosphate if we added a third. So again, we have these monophosphates, and so to make a polynucleotide, so to make a piece of DNA or a piece of RNA, we actually make phosphate esters on both the 3' and the 5' carbon. So we call them 3' and 5', not to be confused with the carbon numbers in the base. So if you hear me say 3 and 5, if you hear me add the prime, we're talking about the sugars, the numbered sugars in the sugar, or excuse me, the numbered carbons in the sugar, not in the base pair, because those also have nitrogen. So what we have, though, when we start connecting these, this oxygen here, as you can see it's better drawn in your book, is connected between the oxygen from the 3 to the 5. So we have one of these linkages here, right, of the phosphate connects both onto carbon number 5, but also down below to carbon number 3. So this linkage here that we have that goes between, and so now here would be carbon number 5 at the bottom, we have a 3 and a 5' linkage. So we see that same linkage between them repeated over and over and over again in the same type. So similar to how we talked about disaccharides or how we talked about peptide linkages. When you have a polymer of this, like a polysaccharide or a protein, it's the same repeating pattern over and over again. It just so happens in DNA and RNA, it is the 5' carbon connected to the 3' carbon, and those are the linkages where the phosphates line up. So as you can see in the slides right here, we have here a free 5' end, that is the start, and then if you notice we have the 5, and then we have 3, we connect O to P to O on the 5 carbon, and then we follow that to the 3, to the O, to the P, to the O, to the 5. 5, 4, 3, O to the P to the O, etc., all the way down. So in DNA, there is always linkages of a phosphate between carbons 3' and 5' in the ribose or deoxyribose, depending on whether you're talking about DNA or RNA. And this actually gives us what we call directionality. So at the start or the top, you will have a free phosphate on carbon number 5. At the very bottom of your DNA or RNA, you will have a free OH group on the 3' end for the two ends. Similar to how we talked about in proteins that you had the N terminus and the C terminus. In DNA, you have a free 5' phosphate, so the phosphate sticking out the 5' end, and then a free OH group on the 3' carbon. Okay, so again, what we have is the linkages between are sometimes called a phospho, so a phosphodiester. So di meaning two, there's two esters on either side. So if you notice with the phosphate, if we pick the one out here in the middle, we have a phosphate and it's double bond O and there's phosphates on either side. So that is a diester that you have. So a monophosphate diester. You might also hear it as a phosphodiester bond. That is what links each individual nucleoside together. So if you take a look, we can use nucleotides not only for DNA and RNA, but just like we saw with ATP. You might have heard of ATP before. ATP is sometimes also known as the cellular energy currency. It's what we use for energy. So again, it's very similar, except instead of being in pieces of DNA and RNA, it's just basically the adenosine base pair with 3 phosphates linked together, just like you can see in the slides here with an adenine base, a ribose sugar, and 1, 2, 3 phosphates linked together. It is a nucleotide triphosphates. So again, we can use these as signaling molecules. Maybe you might have heard of cyclic AMP before. AMP, adenosine monophosphate, okay, or ADP. All of these can be used for signaling, energy currency, a bunch of different things. So here's why we use ATP for energy, is because thermodynamically, if you remember your thermodynamics back from general chemistry, if you cleave off that terminal phosphate, that last phosphate, the products are much more stable. And they're much more stable to the tune of about 30.5 kilojoules per mole. Now that number might not mean anything, but basically that's a big number when it comes to how much energy we can get by cleaving bonds in biochemical systems. So if you cleave that, what we call phosphate bonds, that gives us a lot of energy as a result. So finally, the last thing that we will talk about in the last set of videos, we'll be talking about DNA structure when we put it all together, but then also finally talking about how we replicate DNA and the important pieces for that.